Good morning, everyone, and thank you for joining us. My name is Takudzwa Comfort Madzivanzira, and I'm a PhD student based here at SAIAB. So today it is indeed a special day, or should I say, an emotional one for some of us, especially me and Casey, as we receive a presentation from a person who has been our supervisor. She's been our mother, she's been our sister, she's been our friend here at SAIAB. It's indeed an honor to be hosting this, although I had to impose myself to be the host, and I was ready to put up a fight if anyone else was going to take this opportunity away from me, as the person who is presenting, I just described to you who she was to me. I don't want to say much about her because I might preempt her presentation, as I have a lot to talk about her, and it will end up being my presentation, and I don't like stealing shows. So today we're going to be listening to an exit presentation from a person who is about 1.6 meters tall. She's from England. She holds a BSc and a PhD in marine biology from Queen's University in Belfast, Northern Ireland. After her PhD, she joined the most chaotic group led by our dear late father, Professor Olaf Weyl, that is the Inland Fisheries and Freshwater Research Group at SAIAB. So her research revolves around predicting the impact of invasive species, river restoration, fish community ecology, and searching the lengths of Southern Africa for crayfish. I wouldn't say Southern Africa only, because knowing the capabilities of the person that I'm talking about, she can turn the entire continent upside down just to look for crayfish. She did quite a lot of work, and you don't have to dig much on what she did during her work at SAIAB, because you are all staring at one of her products, that is me. At SAIAB, she supervised me, Casey, Ntombi, and immeasurably and unconditionally assisted most students, not only in our research group. Some of the work that we did in our research group, it was made easy through her links in South Africa, regionally and internationally. So during her time at SAIAB, she authored and co-authored more than 15 peer-reviewed publications, and there is still a lot of papers with her name, which have either been accepted, under review, and yet to be submitted. And I know I'm not the right person to say this, but today I'm the host and I can say whatever I want. I can safely say and declare that her postdoc journey at SAIAB has been a success. Friends and colleagues, the person that I'm talking about is Dr. Josie South, and we're going to be listening to her presentation, and Casey will host the question and answer section, and just feel free to raise up your hand and ask any questions.
Thank you, Mr. Madzivanzira. Hi, good morning. Thanks for tuning into my exit presentation. I hope to share with you some highlights from my time at SAIAB, and I assume most people tuning in will know me in some way or another, but for those who don't, I guess you will by the end of this. Please enjoy. Started at SAIAB in 2018 after a 10-minute interview with Olaf, where the whole thing revolved around the question, do you like fish? So since arriving, it's been a bit of a whirlwind, and I suppose I've only really had time to sit back this past year. Some quick statistics about the last 3 years. They've comprised of 2 postdoctoral fellowships, resulting in 17 publications. I've had the pleasure of supervising 9 wonderful postgrads, managed to complete field and lab work in 7 different countries, been awarded 1 major research grant, hosted 2 international workshops, and presented research at 12 international conferences. During this time, we've almost been mistakenly deported at least 3 times. I've been regularly mistaken to be an honours student, which gets me out of many tricky questions. We set over 1,000 crayfish traps and spent rather little time actually in the Institute. Rather than giving a heavy scientific talk, I'd like to share with you some backstories of some of our published and soon to be published research. There are so many people who have contributed the whole experience and in many different ways, and it's these people I'd like to also recognise today. SAIAB also has a policy of doing presentations whenever we get back from international trips. and I believe I owe a little bit of debt for these, so I hope this fills in some of the gaps. The first thing I was told to do was go on book flights to Cape Town. You're going to the Cederberg with Casey and Angus. And at the time, I didn't know what a Cederberg, a Casey, or an Angus was, and I suspect the whole thing was an initial pressure test. This is the Cederberg, and this is a Casey and an Angus, all of which are excellent. Working on the Rondegat recovery project has been a real privilege. Predatory fish were removed from the lower reaches, and as a result, has stimulated the population recovery of three threatened endemic cyprinids, whose recovery we've been monitoring and documenting to understand the factors which support the persistence in the stream. It's been the most relaxing fieldwork of the entire time. Been hiking through beautiful scenery to set underwater cameras twice a year. And a bonus being that there are very few things that are likely to try and eat you. Saying this, Olaf promised us that you never see snakes in the Rondegat, which really did please Taku, who absolutely hates them. Although 2 days later, after this conversation, a very large puff adder came sailing down the middle of the river while we were busy retrieving cameras. However, both Casey and Taku were too busy, one intent on filming the snake and the other intent on getting as far away from the snake as possible, to remember to shout around the river bend and warn me until the very last moment, at which point the only escape route was to dive into a bush. Casey's been doing some really lovely work as a result of this, characterizing the habitat associations of the redfin minnows and Clan William yellowfish. We found that the fiery redfins are restricted to the highly vegetated upper reaches, whereas the yellowfish are associated with the deep pools and more disturbed lower reaches, although they're probably constrained by water depth, and both species are at risk from drought and sand slugs. He's also been doing some really neat ecomorphology and trophic interaction analyses on 3 species, to help understand how the very similar species can coexist in a resource-limited and range-restricted area. And he's found evidence for both niche separation and habitat filtering. This work's hopefully soon to be published in Aquatic Conservation. A really big initial highlight was the SASEX conference in 2018, and I think all who attended probably agree with me here. Couldn't not mention it, as it was my first real experience of being part of the SAIAB community, and it culminated in the now infamous party in room 57. So during SASEX, 2 important events happened. First being the dysfunctional ecology workshop, which, despite playing out like a disturbing mix of the films 12 Angry Men on a Beautiful Mind, it has been a springboard for some large and ongoing collaborations in invader impact prediction methodology. The second was being told, arguably far too late in the evening, to go find Taran and Victor. We've got this cool camera thing, and you should persuade them to let you use it. And somehow Taran and Vic did let me come and play with the behavioural tracking system. And this brief interaction has led to a really wonderful ongoing collaboration and friendship. Managed to escape Grahamstown for a week to work with Dr. Taryn Borter and soon to be Dr. Nicole Vormerans, who kindly lent me his precious frogs to work with. We managed to thrash out 2 neat papers which were published in Ecotoxicology and African Zoology. The first looked to understand how environmentally relevant levels of DDT might be affecting trophic interactions between the African clawed frog and mosquito larvae prey. We found that DDT had differential effects depending on the species, and that both foraging and anti-predator responses were dampened by exposure to DDT. Second quantified Xenopus behavior responses towards different prey cues and found that olfactory pheromone cues can strongly stimulate foraging behaviors, which indicates how Xenopus can locate food in turbid environments. Another outcome from SASEX was the follow-up workshop led by Dr. Leonardo Kirker, where we dissected and measured an awful lot of fish in a room with no sinks, no water, and no drainage, which in retrospect was perhaps not the best venue choice. The workshop was designed around relating form with function in various fish species and using a variety of measurements to predict species diet specializations. Leo has since been quite integral in some of mine and students' work, and here's some outputs from our recent paper in Biological Invasions. Here we used internal and external measurements to predict and explain how largemouth bass are able to cause so much ecological damage compared to the native Cape Kerper. You can see bass here in brown. It turns out that the bass are far superior fish pursuit hunters, which gives them an advantage in simplified habitats, whereas the Cape Kerper are more specialized for ambush tactics, which rely upon complex habitats and are unfortunately now far less common. After a good few months of being looked after and made to feel at home at SAIAB by Dr. Dumasani Kosa and Dr. Lubabalo Mofu, In September 2018, it was my turn to drag them away to the cold of Northern Ireland to return the favour and share with them a little bit of my home. We spent a month at Queen's and we did a lot of functional response experiments on things with pincers. We found that the globally invasive red swamp crayfish had a considerably higher predatory pressure on both benthic and pelagic prey, regardless of substrate. We found a surprisingly high consumption rate on Daphnia, as the crayfish seemed to create a flow to draw the Daphnia closer so that they could be eaten. We also completed some experiments on the commercially important Nephrops norvegicus to understand prey consumption with regards to size class and prey species. This is currently being written up by PhD candidate Monica McCard. The majority of my time has, however, been devoted to coordinating the Pan-African crayfish hunt, as well as Mr. Madzivanzira's PhD project. Red claw crayfish have established throughout the Zambezi Basin, but until now, the efforts to locate and assess their extent have been disparate and not standardized. These prior works allowed us to review where the crayfish have been reported from and use these localities to plan an extensive standardized survey as part of the first strategic assessment of the invasion, as well as determining critical knowledge gaps in which we would try our best to start filling in. So soon after we returned from Ireland, it was time to start the surveying. Myself and Taku prepared to go to Lake Malawi to help Angus finish up his camera surveys, where he was assessing the chumbo fishery in the lake. We were then to drive with him in the trailer to Lake KARIBA to finally start the big fat Pan-African crayfish hunt. Being at the lake was like spending 2 weeks in a dream. Lots of hauling BRUVs out of the depths of Lake Malawi, watching Taku teach himself to swim, and a healthy dose of Malawi gin. While we were here, we also had a meeting with the Monkey Bay Fisheries Department to discuss the importance of monitoring the Shiri tributary of the lake, as this is the most likely route for any incipient crayfish invasions. When we got to the University of Zimbabwe, Lake KARIBA Research Station, me and Taku were really excited to finally get going with data collection to compare gear detection rates and CPU upcalculation rates. Angus, on the other hand, was very excited to not have to sleep in the tent for the first time in 3 months. While we were there, Olaf texted us to say, go visit my parents, and he sent us an address. We decided we would before we left. This was in December 2018, just before the next round of political unrest and period of severe fuel shortage in Zimbabwe. This wasn't really conducive when we had to account for both boat and bucky fuel in order to get our data and get home. We were busy throwing traps from the shoreline of the lake. We were very tired and grungy from working all day. We could see someone watching us really quite intently. While we all enjoy talking about work and what we're doing, sometimes the last thing you want is to be interrogated by a member of the public, especially when you're covered in dirt. So we're all a little bit tense when this guy comes up to us and announces in a very big German accent, "So I guess you must work for my son." Spoiler, we did work for his son. Uli and Gabby kept us really entertained and fed that evening and helped us find enough fuel to complete the work and make it back to Johannesburg. Although it did turn out that Olaf had given us the completely wrong address for his parents in the first place. Our work completed here was accepted for publication this week and gives the upcalculation factors for the disparate gear types used for crayfish sample in Southern Africa. This was a really essential first step in order to not make erroneous conclusions based on the CPE of different methods. The Zimbabwean opera traps and the collapsible Promar traps both caught similar length distributions and sex ratios of crayfish. However, the detection probability and CPUE of the opera traps was significantly lower, which meant that the historic data calculated from the lake needs to be upcalculated by a factor of 2.67. In March 2019, I was awarded European Union funding in order to take a team of South African scientists to the Netherlands to spend a month and see if we could figure out what happens to trophic interactions when exposed to fluoxetine, the active ingredient in the antidepressant Prozac, and microplastics simultaneously. While we were here, I think we went a little bit mad from vitamin D deprivation after working rather intensely in a windowless lab. And by the end of it, we could have all probably done with a little bit of that Prozac. But we did get some cool results, which are currently under review in Science of the Total Environment. Our results indicate that neither of the pollutants cause invertebrate trophic interactions to change significantly from the control, although microplastics did slightly decrease the feeding rate of the model predator, and fluoxetine alone slightly increased the feeding rate. However, there was an antagonism when exposed to both microplastics and fluoxetine, which cancelled out the two effects and resulted in feeding rates similar to the control. These effects were driven by species-specific effects on swimming speed and distance. In particular, the microplastics alone caused the daphne to swim far faster than the Calboris did, meaning that the decrease in feeding rate is due to the Calboris literally not being able to catch their prey. When we got back from the Netherlands looking a little peaky and worse for wear, I was told, "Don't worry, South, I'm going to send you on stress leave to the Okavango Delta." The real reason was the first leg of surveying the Upper Zambezi to locate the invasion front and invasion core of red called Crayfish. Craig, Taku and myself drove Sparky through the Kalahari Desert and into the beautiful Zambezi region. We did have a little bit of a hitch on the way though. Our permit papers weren't quite correct due to a recent change in legislation, and we'd taken a wrong turning coming through the very small Onsiepkans and Velos drift border. The border guards told us that they'd give us 10 days on our passport, but we had to go immediately to Home Affairs in Vin to complete our case. We saw a lot of really amazing things in Namibia, but easily the most impressive was during our night in a very mediocre Keepman Soup campsite before we made it to Windhoek. Myself and Craig had been struggling all night to put airtime on our phones and set up Namibian SIM cards. It was really quite cold, as you can see here, and we assumed Taku had gone to bed until we heard quite a lot of shouting from his tent. Turns out he had somehow managed to not only get airtime, but also a DSTV uplink to watch the Liverpool match live and hotspot it from his phone in the middle of a desert. And frankly, I don't think I've ever felt quite so incompetent. The hitch with Home Affairs was sorted after a brief mistaken identity, where we tried to speak with the minister. We'd been driving all day and run straight to the office before the end of the day, and the minister happened to mistake us for backpackers. So the next morning we put on shirts with logos and some long pants and went and reintroduced ourselves. At this point there were a few, "Oh my God, I'm so sorry, I didn't realize you three were the scientists people kept bringing me about." Luckily it was fixed in a matter of hours. And if we look a little bit harassed in these pictures, because someone was purposely flicking a fishing line over our heads while we were trying to find our traps and not get stuck in the sandbanks. A lot of the time was spent shoving dog food into stockings in very scenic locations, dodging the ridiculously abundant hippo population of the Okavango River, camping on sandbank islands, and that one weird week where we lived in the car park in Katima Malilo. During this surveying period, we set 358 traps and didn't catch any crayfish. This confirmed absence, or at least populations below detection level in the Okavango, Pondu, Zambezi, and Chobe rivers. We did, however, catch a lot of Synodontis, Potaminotic crabs, and Craig proved himself to be the tigerfish whisperer. The big fat crayfish hunt continued again in the low water season, this time in Zambia, starting in the Kabompo River and joined by our friends, the parasite ladies, Marlies and Alini. Owe some thanks to this entire group, actually, for their continued patience and kindness with my kind of hectic malaria tablet-fueled sleepwalking night terrors every night. So much to the fact that after 2 weeks of nighttime screaming, they had collectively managed to condition my sleep self to politely ask, "Hello, is my tent floating away? Please can I come out?" And instead go back to sleep after being told, "No, Josie, you're fine, you're not floating away." So Marlies and Alini were a really fantastic addition to our weird, unlikely trio. Made us continuous buckets of popcorn, did lots of field haircuts, and for pouring formalin over my burnt and bleeding feet in a last-ditch attempt to stop them from getting so infected after I walked through the previous evening's fire, all the while sifting through every inch of catfish looking for very tiny parasites. In order to find enough catfish for the parasite ladies, we paid a local fisherman to catch catfish alive, which was a disaster in its own rights, as well as staying up late fishing for them every night. One morning, we had headed out early after an unsuccessful night of fishing. Myself and Craig had taken the nets and gear to the pontoon, and Taco had gone to bring the boat round from the mooring. We thought it was taking his time a little bit, until from around the corner there was some loud yelling, "Craig! Josie! Craig! There's loads of water in the boat." Shouted, "Yes, Taco, you know it's a wet deck, it's fine, hurry up." "No! There's too much water." So we dropped the kit and ran around. Sure enough, Sparky was almost vertical and full of water. We started to panic a little bit and really wondered whether we'd somehow put a hole in her without realizing, but we bailed the water and luckily the engine hadn't flooded. She ran completely fine, so we carried on with our day, only a little bit confused. But after a few hours in the sun, the boat was stinking, and we realized that there was a bag of fish bait left forgotten overnight in one of the alcoves. So we came to the only feasible conclusion that the boat was full of water because one of the unreasonably large Kabompo crocodiles had smelt the fish and tried to haul itself into the boat and managed to instead sink half of the boat in the process. After leaving Craig to finish his microhabitat sampling in Javundu, parasite ladies left for Mongu, and myself and Taku set out to sample the length of the Brotsi floodplain. We covered the 214-kilometre stretch from Lukulu to Sanango, looking for crayfish and taking isotope samples from the entire food web across the invasion gradient. In Lukulu, we were almost arrested and deported twice by our friends in the picture here, but it was only a simple mistake, and they instead took pictures with us and left us be after inquiring where exactly was the blonde South African boy they'd seen the last time. So the only conclusion is that Craig is always getting us into trouble. A really wonderful memory from this leg of the trip was giving a presentation and having a chat with the Lukulu Secondary School Leavers class outside beneath the shade of this massive tree. We managed to confirm the presence and extent of the invasion in the Brotsi floodplain, with the invasion core found centered around Mongu. With support from the WWF, University of Zimbabwe, Namibian Ministry of Fisheries and Marine Resources, and the Zambian Ministry of Fisheries and Livestock, we managed to map the extent of the crayfish invasion throughout the Upper Zambezi, altogether surveying 10 rivers in 4 countries. We found that the rate of spread throughout the Brotsi floodplain is around 49 kilometers a year downstream and 12 kilometers a year upstream. In terms of abundance, we found that the most recent invasive population in the Brotsi floodplain has not yet reached the full extent that the populations in KARIBA and Kafue have. We might be able to use the older invasions as a forecasting tool. This monumental piece of work is currently under the second round of reviews in aquatic conservation, and hopefully we're able to share it soon. A really interesting facet from the distribution abundance surveys was the high proportion of intersex individuals present in all 3 invasion cores, with the most recent Brotsi floodplain invasion being around 8% intersex individuals. This means that while they behave like males in terms of territory and fighting, the intersex individuals breed as females and produce offspring which are disproportionately female, which might be contributing to their invasion success globally. Our review paper had identified huge gaps in African crayfish invasion impacts, and more recently a global review showed a data deficit specifically for the red claw crayfish. A series of lab experiments, which are either published or soon to be in review, show that the red claw crayfish has the potential for high predation on juvenile African sharp-toothed catfish, regardless of temperature. It's the blue line here. Another experiment aimed to quantify scavenging rates of invasive crayfish compared to native crabs, and allowed us to calculate the consumption per gram of crayfish, and then use this to predict maximum scavenging rates in the 3 invasion cores. Given that the Brotsi floodplain population is neither as abundant nor as heavy as the Kafue or KARIBA populations, it's highly likely that the invasion impact in the Brotsi will continue to increase to the same levels as in Kafue and KARIBA. I contributed to the recently submitted INVACOS project on crayfish economic costs. There are no data for economic costs of crayfish for the entirety of Africa or globally for the red claw crayfish. We've been able to combine our lab and survey data with the WWF's fisheries assessments in the Kafue and Brotsi floodplain, and I have very good reason to believe that between Mr. Madzivanzira and Mr. Nawa, we'll be able to contribute the first quantitative data on crayfish economic damage to fisheries in Africa very soon. All of this work has been done with the support of WWF Zambia, the Upper Zambezi Landscape Project, a project which was far broader than the aspects mentioned today. This photo shows many of the project partners in one of our last in-person meetings before the pandemic set in at SAIAB in January 2020. Besides messing about in swamps far from home, there has been a fair share of surveys and workshops based in the beautiful South African national parks. We've been looking for largemouth bass and redfin minnows in Addo to understand how drought facilitates or impedes invaders, and holding impromptu workshops in the Kruger with the esteemed professors Britton, Dick, and Ricciardi, all of whom have been hugely supportive and crucial in my research and career, both before and after the loss of Olaf, and especially in making sure that I've had something to do in the last year while stuck in the UK. My final jaunt before my visa debacle should have been the pièce de résistance, but obviously it ended up being utterly disastrous. A meticulously planned year-long field experiment to test the leading-edge hypothesis in sharp-hoofed catfish was scuppered, when it turned out that people had obviously got word of there suddenly being tons of catfish in the irrigation ponds, and instead gone to fish them out and eaten, the majority of the 300 transplanted fish. Weirdly, this did end in some major team building on the last day when Olaf appeared with half of the lab group and every single fight and gill net in the garage, while insisting, were we sure we'd been setting the gear right? I think that evening we set around 120 meters worth of gill nets and 5 fight nets in one pond, resulting in a fish catch of about 12 fish, none of which were catfish. So we called it a day with a few tubs of ice cream, but what was particularly rewarding from this was after 2 years of me doing all of the extensive fieldwork driving, Taku finally having his driver's license and getting to be chauffeured by him instead. There have been far too many people, both in and out of SAIAB and in and out of work, who have made my time here as special as it has been. And it really is thanks to all of you guys, especially those of you, and you know who you are, who repeatedly rescued me from my constantly broken down car, taught me just enough Xhosa to greet Sislu in the mornings and say, "Pake mali uzobono," which might come in handy for grant applications, but I remain unconvinced. And finally, for never ever waking me up during my lunchtime nap, even if PhD students are looking for me. I feel really lucky to have been part of this, and it doesn't really feel right to be doing this presentation from the UK, and especially not without Olaf, without whom all of this wouldn't be possible. It's a really rare situation in research to turn up and be told, what are you interested in? I've got loads of money, so you can do what you want. As a result of this attitude, I'll be leaving this position with a network of friends and colleagues spread across the globe, many wonderful memories, and realistically some sort of intestinal parasite. I hope you enjoyed hearing some of the many stories behind the paywalled articles, and I'm sure that there'll be more to come. Thanks very much.
Thanks, Josie, so much for that great presentation and sharing with us all of your awesome experiences, and it's been awesome to work with you over the last few years. So now we'll be moving into some question and answers or any comments that people may have. So if you want us to read out your question and Josie to answer it, please put it in the Q&A box. And if you want to ask a question live, please raise your hand. So we have a comment here from Sarah Davies. Just a comment, it's great to hear all of the details of the findings from Josie's work. There was a lot I wasn't aware of through Josie's SAIAB fellowships. And I don't know if Josie wants to say anything on that.
This has been kind of non-stop, so there wasn't ever too much time to give feedback. So it's really nice to give this presentation today and actually share a little bit of it, because I think lots of people just got lots of obscure fish references via social media. So it's good to properly talk about it.
Okay. Matt Parkinson says, my poor boat.
I'm sorry, Matt. I blame Taku, honestly. It got treated very nicely. We never put a hole in it.
Okay. Jessica Glass says, great job, Josie. What is the connection again between the Prozac fluoxetine and the microplastics?
So they're both very, very common and co-occurring in pretty much all waterways. The idea and what we got a little bit of evidence was that they might interact with each other. So the fluoxetine is the active ingredient in Prozac, and it looks like it might bind to the microplastics, which then makes it less bioavailable in the rest of the water column. However, we're not sure whether this then makes a difference for things that eat the microplastics directly, as they suddenly get microplastics and a lot of Prozac, whereas other things that don't eat the microplastics directly might not be affected so much.
Okay. Leo's Nagelcoco says, lovely talk, Josie. Incredible how much you've been doing in this short time, and I agree. And then further, he asks, are you going to continue with the crayfish work?
Hey Leo, yeah, hopefully. I'd really like to continue with the crayfish stuff. I think there's lots to be done, especially still in Africa. So I hope that the boys are going to continue with it, but we'll see.
Taryn says, it's been an absolute blast working with you, South. I'm sure we will continue to do so in our careers going forward.
I hope so, Darren. Thanks.
And Dennis says, great talk, Josie. When will we see you again?
Dennis, I'll probably see you in England before I see you in South Africa, the way it's going. But hopefully, maybe December, we'll see what happens with my visa debacles.
And then Matt and John say, what's next and where to next for Dr. South?
Well, I think John already knows, but for the rest of my contract, for now, I will be adopted by Professor John Measy at SAIAB in Stellenbosch, and hopefully get to play a bit more with some frogs, which I'm quite looking forward to.
See if there are any raised hands or any further questions.
Looks like us.
Right. Yeah, that looks like it. But yeah, thanks again, Josie. Awesome presentation.
Thank you, Casey.
Yeah, thank you. And yeah, I'm sure we'll see more of you. And yeah, you'll be moving down to the Cape where I am. So sure, I'll see more of you as well. And yeah, we'll just make sure that December party happens.
It definitely will. I think we're owed some celebrations around. Thanks very much. And thanks for tuning in, guys.
Okay, thank you very much, everyone.